Welcome to the Explainer. If you're a focused learner who really wants to get into the nuts and bolts of how the human heart actually works, well, you're exactly where you need to be. Today we're looking at the heart, not just as, a basic anatomical organ, but as this absolute marvel of biological engineering, a totally self-regulating machine.
Okay, let's just dive right into this. Think about this for a second. Your heart is roughly the size of your fist, right? And it weighs only about 10 ounces. But this relatively tiny muscle pumps out about 5 liters of blood every single minute. We call that your cardiac output. And the crazy thing is, when you go for a run or hit the gym, that demand spikes, and this tireless little engine just instantly ramps up the pace to match it, no hesitation.
So how exactly does it do that? Well, here is our roadmap. We're going to hit 5 key areas today. First, the anatomy of the heart. Second, the 2 circulatory systems. Third, the cardiac cycle explained. Fourth, the Frank-Starling law. And finally, number 5, the cellular engine. Basically, we'll start wide with the macro stuff and then zoom all the way in to the microscopic level.
All right, let's kick things off with part 1, anatomy of the heart, the rooms and doors.
The absolute easiest way to visualize heart anatomy is just to think of it like a building. So the thick muscular walls, the myocardium, they house 4 distinct rooms. Those are your chambers. Connecting these rooms, you've got 4 doors, which are your atrioventricular and semilunar valves. Then, of course, you need plumbing. That's your blood vessels, with the arteries carrying blood away and the veins bringing it all back. And finally, running the whole show is a highly intricate electrical wiring network. That's your conduction system, firing off signals from the SA node at the very top, all the way down to the Purkinje fibers, telling the muscle exactly when to squeeze. And when we look at the structure here, notice how the whole building is basically split right down the middle by a wall, which we call the septum. The right side of the building, so the right atrium and right ventricle, deals exclusively with oxygen-poor blood coming back from your body. Meanwhile, the left side, the left atrium and ventricle, that side is entirely dedicated to handling the freshly oxygenated, rich blood coming straight back from your lungs. It's a continuous, strict 1, 2, 3, 4 journey. Step 1, that oxygen-poor blood flows into the right heart via these massive veins called the vena cava. Step 2, the right ventricle pumps it straight out to the lungs. Step 3, the lungs do their thing, reload the blood with oxygen, and send it right back into the left heart. And finally, step 4, that incredibly powerful left ventricle squeezes and launches this rich red blood out through the aorta to fuel your entire body. Which brings us to part 2, the 2 circulatory systems, a tale of 2 pressures. Now, because the right and left sides are shipping blood to totally different destinations, they have to operate under drastically different physical conditions. The right ventricle is just pumping next door to the lungs into the pulmonary circulation. So it's a very low-resistance, low-pressure system. A normal pulmonary systolic pressure is only around 18 to 25 millimeters of mercury. Pretty chill, right? But the left ventricle? It has to pump blood into the systemic circulation, which means the rest of your entire body. That takes massive force. We're talking high resistance, high pressure, with a normal systolic pressure of about 120 millimeters of mercury. It's a completely different ballgame. Moving on to part 3, the cardiac cycle explained, cause and effect. So every single heartbeat is governed by one fundamental rule of physics. Blood only flows down a pressure gradient. High pressure to low pressure, always. The cardiac cycle itself is basically 2 main states. You have systole, which is when the heart muscle actually contracts and ejects blood, and then you have diastole, which is when it relaxes and fills back up. Now, when that muscle contracts during systole, the pressure inside the chamber skyrockets. And once that internal pressure finally pushes past the pressure on the other side of a closed valve, it quite literally forces that biological door open, and boom, blood is ejected. And if you want to see exactly how these pressure shifts play out, the famous Wiggers diagram charts it perfectly. If you trace the relationship between left ventricular pressure, which is the yellow line here, and aortic pressure, the red line, you see something amazing. During contraction, the left ventricular pressure shoots straight up. But notice, it's only when that yellow line crosses above the red line, meaning the ventricle's pressure is finally actually higher than the aorta's, that the aortic semilunar valve is forced open and the blood rushes out. So the whole cycle really breaks down into a few key phases. First, you've got ventricular filling during that relaxation phase. Then there's this split second called isovolumetric contraction. The ventricles are squeezing hard, but all the valves are still closed, so the pressure just shoots through the roof. Then comes ventricular ejection. And finally, isovolumetric relaxation, where the ventricles chill out, the pressure drops, and the valves snap shut.
And actually, you know that classic lub-dub sound you hear through a stethoscope? It's kind of wild, but that's not the sound of the muscle squeezing. The lub, or S1, is actually the atrioventricular valve snapping shut, and the dub, S2, is the semilunar valve snapping shut as the blood tries to fall backward. All right, let's hit part 4, the Frank-Starling law, the heart's automatic transmission. Okay, so we know the heart pumps out blood, but here's a really interesting question. How does it know how much to pump out at any given moment without your brain constantly micromanaging it? Well, that's all thanks to the Frank-Starling law. This law dictates that your stroke volume, which is just the amount of blood pumped out in one beat, increases in direct response to an increase in the volume of blood filling the ventricle right before it contracts. We call that pre-contraction volume the end diastolic volume. It's exactly like stretching a balloon, right? The more air you blow in, or in this case, the more blood that flows in and physically stretches the heart muscle, the more forcefully that muscle is going to snap back and contract to push it all out. And the reason this matters, the real genius of this physiological mechanism, is that it perfectly and completely automatically synchronizes your right and left ventricular outputs. Think about it. If your right heart suddenly pumped way more blood into your lungs than your left heart could pump out to your body, blood would rapidly back up into your pulmonary circulation, and that would literally be fatal. But the Frank-Starling mechanism ensures that whatever volume enters the heart is perfectly matched by the volume pumped out. It keeps your whole system beautifully balanced all on its own. This leads us to our final section, part 5, the cellular engine inside the sarcomere. So how does that physical stretch actually create a stronger contraction? For that, we have to zoom way in on the striated muscle cells. Muscle contraction relies on what's called the sliding filament theory. Inside the basic functional unit of the muscle, the sarcomere, you've got two main proteins, actin and myosin. When triggered by a sudden flood of calcium ions inside the cell, the myosin heads form cross bridges and literally reach out and pull the actin filaments toward the center. This microscopic pulling action, fueled by cellular energy called ATP, is what physically shortens the muscle and creates a heartbeat. Now, unlike the skeletal muscles attached to your bones, the heart muscle's resting sarcomere length is incredibly variable. It totally depends on how much blood is filling the chamber at that moment. But here is a super precise, fascinating detail. In a human heart, the absolute maximal force of contraction is generated when that initial sarcomere length is stretched to exactly 2.2 micrometers. That's the sweet spot. Back in 1963, a cardiologist named Edmund Sonnenblick used electron microscopy to figure out exactly why the Frank-Starling law works at this microscopic level. He discovered that as extra blood flows in and stretches that heart wall, it physically decreases the gap between the actin and myosin filaments, and because they're closer, a lot more cross bridges can form. But wait, it gets better. This stretching actually increases the sensitivity of a protein called troponin to calcium. So a purely physical stretch directly alters the molecular biochemistry of the cell, giving you a massively stronger pump. From those large, high-pressure, muscular rooms all the way down to the microscopic, calcium-triggered sliding filaments, your heart really is an absolute marvel of biological engineering. I mean, it tirelessly regulates its own output, adapting beat by beat to whatever your life demands of it. So, knowing just how flawlessly this 10-ounce automatic pump adapts to your every need, the question I want to leave you with today is, how will you make lifestyle choices today to protect your own plumbing and wiring for tomorrow?